So our next presenter is Dr. James Schnaps, and I think when I read his bio, I just say, "Wow, wow!" So I tried to find four keywords to express, kind of describe him. The first one is a researcher, as we can expect, a big researcher and a lecturer, of course. The second is an entrepreneur, and you will see in the next few lines why he's an entrepreneur. So it means that even as a scientist, we can still have businesses. And I think we need money to run research, definitely. And when we have good research, we need to commercialize it, a good connection. And fundraiser because raise more than 20 million USD. It's not an easy task. And last but not the least, an award winner. Of course, when you are doing the good things, you win awards. And so in the next couple of minutes, go through his background in a more detailed manner. Okay, so Dr. James Schnabs, yeah. I think I introduced you very well, and now the whole things disappear. Okay, all right. So I think we can stick to that with the introduction. Everything else is more of details about his background. He's from Nebraska, Lincoln, and I think as we are talking about connection, about networking, this is a good opportunity not only for us to learn from him, from his research activities, but also to reach out to him. He may be interested in taking some of you on board and of his research program. So, Dr. James, you have the floor, and the floor is yours. Over to you.
Yeah, thank you so much for that kind introduction, and thank you so much for the invitation to be here.
Thank you.
It's the second time I've had the chance to attend the West African Symposium. And just to establish expectations, I remember from last time that the questions I got were outstanding. So I'm expecting similar fascinating questions today. Let me share my presentation here. Is that coming through okay? Good. All right.
It's okay, yeah.
Yeah. So today I'm going to be telling you two very different stories, both of which are on the topic of how we are going to address the ongoing need for more food around the world and doing that in an era where it's getting harder and harder to advances in plant breeding. And it's getting harder and harder to ensure that we're going to have optimal conditions to grow crops in. Before I get into that, the university I'm at really wants me to disclose my conflicts of interest. So I have been involved in founding three different companies, Ingenious Ag, Dryland Genetics, and Data to Bio. I'm going to be telling you a lot about Dryland Genetics in the first part of my talk, which is a company that is breeding not a new crop, but a crop that has not benefited from a lot of the modern plant breeding technologies that we have access to today. I also serve as an advisor to a number of other startups. And about 2 years ago, I spent some time as a paid employee of Google. I'm also an inventor of a patent that covers some of the technology for understanding genomic sequence that I'll cover later in the talk. And I'll point out where that part is, because again, it creates a potential conflict of interest. And when you see me present about this work, you should keep in mind that I'm also one of the inventors of it. With that, an important thing we have to remember, and this is a slide I start up almost all of my talks with, is that when people are not able to afford to eat, the world becomes unstable. What you see here is an FAO food price index chart tracking food prices going back over the last 3 decades. I grew up in an era of very stable and reasonably low food prices in the 90s and the early 2000s. As a researcher, I've worked in an era where the price of food can spike much more dramatically. And when food prices go up, when people are confronted with whether or not they're going to be able to afford enough to feed their families, they tend to lash out. They tend to push back at systems that are not allowing them to feed themselves. And we've experienced a number of these spikes over the years. So one of our essential jobs as plant scientists, as plant breeders, is not just to feed people. We need to feed people. But beyond feeding people, we are actually ensuring that we and everyone we know are able to live in functional and stable societies. And food prices are continuing to go up again, although this slide is a little bit out of date in the last year or so. They're down, which is good for everyone who eats and makes farmers a little bit discouraged. So you can never make everyone happy. I also like to show this. During the pandemic about 4 years ago, there was a big shift towards online ordering of groceries. And what this allowed me to do is go back into my own personal consumption history. So you're seeing the sorts of foods that a professor in Nebraska tends to eat and cook for himself, and how much those increased over a period of about 2 years. So the increases in food prices can be quite large. So why is this happening? Why are food prices so much less stable and more prone to spiking than they were in the past? A big part of the reason turns out to be something we can attribute to the three grain crops, rice, wheat, and maize, or corn, that contribute more than half of all the calories consumed around the world. Just these three crops underlie essentially our entire ability to support ourselves, to support global civilization, the human population. And large increases in the productivity of these crops, the amount of grain we could harvest from the same amount of land, allowed us to meet growing demand for many, many years. However, as you see here, if we look at many of the most productive regions around the world, the yields, the productivity of both rice and wheat have stopped increasing. They've plateaued. And so as the number of people increases, as people's dietary preferences shift so that they are consuming more calorie intensive foods, the big thing is more meat consumption. We have more and more demand, and we're no longer able to meet that with rising productivity in these two crops. Now, the third of these crops, corn, the story is still a little bit better. You're seeing here data from the US rather than the world, but you're seeing the productivity of corn in the US going back about 120 years. And you can see that it was stable for quite a while. And then we had two stages of increased two stages of increase in productivity. This first one, some of the early breeding efforts, often using four-way hybrids. And this later one, looking at single hybrid crosses, replicated statistical plant breeding, as well as widespread access to abundant nitrogen fertilizer. Since the 1960s in the US, we have managed to increase corn yields by an average of about 2 bushels per acre per year. And over the course of decades, that is translated to roughly a 6 to 7x increase in the amount of corn that can be produced from the same amount of land. The increases like this are what has allowed everything in agriculture to proceed to today. The challenge is that achieving this same increase, this 2 bushels per acre per year, is a smaller and smaller percentage increase as our baseline yields get higher. And achieving even this constant 2 bushels per acre per year is getting more and more expensive. It's requiring more and more investment. And at some point, it will not be sustainable to continue the large investments in plant breeding necessary to achieve even these relatively slow increases in yield. So like I said, this is how I typically start out my talks, set up the big challenge, the big concern, and then I try and present some reasons for hope. Oops. Yeah, let's skip over that. Here I'm going to talk about two stories, one in my entrepreneurial hat and the other in my academic hat, about ways that we can continue to meet the growing demand for food, continue to meet the need for more resilient crops as weather conditions become more severe and more extreme. The first of these is the new crop story. And this is a story that I did not conduct as a professor. It's a story that I, developed independently of my academic work because it is very hard to actually get funding for real plant breeding efforts in the public sector in the US. On the other hand, if you can make a compelling pitch, it's possible to raise investor money to invest in a 5 to 7 year process of developing new crop varieties and releasing them. But you have to be able to make that compelling pitch that the money that people are putting in today is going to come back usually many times over what they put in 7 or 10 years, while still leaving enough value to dramatically benefit farmers, to benefit community, to benefit food security. In this case, the problem that I set out to address was that agricultural production in the US is increasingly limited by access to water. What you see here is a map of where water supplies are under the highest pressure in the US, both when we look at rain-fed and irrigated corn. We are seeing both a decrease in rainfall in the western edge of the Corn Belt. We're also seeing that as cities grow, they compete very effectively with agricultural land for water. Most of these decisions are made by democratic governments at the regional, state, or national level. Cities have a lot more voters than farmers do. And so the cities win when they go head to head with farmers about who's going to use limited water. And so large parts of the Corn Belt are losing access to the water that they need to grow crops. Oops. Exacerbating this problem in a large part of the western edge of the US Corn Belt, the way that we get enough water to grow corn is we pump it up from an underground aquifer, the Olagala, and this aquifer is drying out. We've been depleting it. We've been pumping out more water than is recharged each year. And in large parts of Kansas and the Texas Panhandle and Oklahoma, the wells are running dry. It's no longer possible to bring up the water that is used to irrigate these large center pivot irrigators that were the reason corn could be grown in large parts of this region. And when we look at how corn yields have changed over time, so I showed you that graph showing that corn yields have increased dramatically at a national level, about 2 bushels per acre per year. The vast majority of that increase is coming from a relatively small part of the corn growing country in the US. This region centered on Iowa, southern Minnesota, parts of Illinois. Now, these are the most intensively farmed parts of the US and some of the most intensively farmed land anywhere in the world. So it's a little bit deceptive. There's a lot of land here, but there's less corn grown in these regions than here in the center. But this is where the seed industry has been concentrating their breeding efforts because they can sell the most corn there. And these are areas that typically are not very input limited. So they have very good soil. They have plenty of water. Farmers are able to apply as much fertilizer as the crop is able to use. But it means that breeding is developing crop varieties that are optimized for these optimal resource-intensive conditions and not optimized for the drier and more extreme environments in the western edge of the Corn Belt. People have spent a lot of time trying to make corn use water more efficiently, tolerate drought more effectively, and they haven't had a lot of success. We now have corn varieties that can wait longer for water to arrive, but to make 200 bushels of corn from an acre of land will take about twice as much water as taking 100 bushels. The efficiency with which corn converts water into grain hasn't changed very much, even as our productivity has increased. So as productivity increases, the amount of water you need for a given field also increases. On the other hand, in the US and the western edge of the Corn Belt, we have this crop called proso millet, which is the focus of my breeding efforts, that is able to grow with far less water than corn. And as I'm going to show you in the next couple of slides, actually can produce more grain from the same amount of water. Now, the yield of proso millet in optimal conditions is lower than in corn, but in conditions like this, where there just isn't enough water, the proso millet produces a crop, the corn does not. Now, proso millet is a grain crop like corn, like rice, like wheat, and you can use it for all the same things. It gets used for human consumption, it gets fed to animals, it gets fed into different processing plants. It's essentially a drop-in substitute for all of the things that we use cornstarch and corn protein for. This is a crop, it was originally domesticated in northern China more than 10,000 years ago, moved with trade routes and cultural exchanges from China into South Asia, the Middle East, into Eastern Europe, was brought over with immigrants to the US about 200 years ago, and is now grown on about 200,000 hectares out here in the western part of the Corn Belt. So why was this crop carried all around the world, and why was it adopted into so many different agricultural systems? The key thing that makes proso millet exciting is that as a result of a rapid life cycle, a very high harvest index, so it produces a lot of grain and not a lot of the rest of the plant, as well as we think some very interesting photosynthetic innovations. Proso millet produces a lot more grain from the same amount of water. It can produce about twice as much, twice, 2.5 times as much grain per unit water as corn. And you can describe that same property different ways. You can say proso millet can grow in areas where other crops like corn cannot grow, areas with maybe only 4 inches. I think there'll be about 10 centimeters of precipitation. In areas that are water limited, it can produce more grain than another crop, even if that other crop can be grown in the same area. Or, for example, in irrigated systems, you could say that it's possible to produce the same amount of grain while using less water, while applying less irrigation water. So this is a very valuable thing, particularly when we are looking at large parts of the globe that are not limited by access to land, but access to water in terms of their agricultural productivity. Proso millet is currently grown on, I think I said 250,000 hectares, primarily in the western US. This represents about 10% of global production. It's also grown in other countries around the world. Relative to other crops, corn, wheat, soybeans, this is a rounding error. This is, less than 1/10 of 1% of total. Maybe it's less than 1%. Yeah, I think that's actually more realistic. It's less than 1% of the land devoted to other crops, but it has the potential to address this key limitation we see in terms of agricultural production. So let's say we were able to convince farmers to adopt this crop. We would need something that would convince them to adopt it. But let's say they've decided that they want to grow the crop. How fast can farmers adopt new technologies, new crop varieties, new crops? If we look back through the historical record, it turns out this can happen really rapidly. In the US, I showed you that there was that flat productivity of corn for a long time, and then suddenly yield started increasing with the introduction of hybrids. The transition from those early varieties, open pollinated varieties, to hybrids in the US happened extremely rapidly. Over a 4-year period, starting in about 1935, we saw the percent of farmland planted with hybrid corn as opposed to open pollinated corn varieties grow from 10% to 90%. Similarly, when we saw the introduction of herbicide-tolerant soybeans, a biotech trait in the US, national acreage of soybeans planted with herbicide resistance traits went from 0 to 75% in about 6 years. We see similar patterns outside the US. For example, the adoption of dwarf wheat varieties that allowed dramatically higher yields in India during the Green Revolution. But again, this is in part an economic pitch. So I want to talk about a specific case of how a very rapid adoption of a crop in the last 30 years. And this is a crop called canola. Canola is a variety of Brassica napus that was previously grown. It made an oil that could be used for industrial applications, but was not suitable for human consumption. Through breeding, it was possible to develop a variety of this crop that produced an oil that was safe and desirable for human consumption. And we saw that over an 8-year period, from 1992 to 2000, the acreage devoted to canola increased 11 times. So again, these things can happen very rapidly. Now, a key thing with canola and a key challenge for the adoption of almost any new crop or substantially different crop variety is that you have to balance the growth of the supply and the growth of the demand. With canola, this was possible because lots of processed food uh, was essentially able to substitute between soybean oil, canola oil, and other oils, depending on which was cheapest. So if farmers grew a lot more canola, they didn't have to worry about saturating a small market specifically for canola oil. Instead, they were growing a crop that could be uh, sold into this much, much larger market of essentially all vegetable oils, all plant oils being used for producing food. And that allowed this very, very rapid adoption. But introducing new crops is very challenging. Um, I imagine some of you have had the experience of working on either new crops or orphan crops that have really desirable properties, whether those are things that benefit the farmer, whether those are things that benefit the person eating the crop. And you have seen just how hard it is to go from zero hectares to 500 hectares, or from 500 hectares to 5,000 hectares. In thinking about this proso millet opportunity in the breeding of proso millet, we identified a number of the challenges here. One of these is simply getting farmers comfortable with growing a new crop. Now, this can mean both expertise in growing the crop, knowing when to plant it, when to harvest it, how to manage it, but also in mechanized systems, often new crops require purchasing new pieces of equipment, which can be very expensive and very risky if you don't know if we're going to continue growing a crop or not. So in thinking about opportunities to develop new crops, new crop varieties, and get widespread adoption, it's really useful if you can select something that can be grown and harvested with existing equipment. It doesn't require large new capital investments. Unfortunately, with proso millet, that was the case. It could be grown and harvested with equipment used for wheat that farmers in our target region already had access to. Similarly, supply chains can be quite challenging. So the farmers who grow this are not all going to consume it themselves. They need to be able to sell it, and they want to be able to conveniently sell it into um, supply chains that will distribute it across the country. In working with a crop that was already grown on several 100,000 hectares, this part was already present. Looking at many, completely new crops, it is discouraging how often that turns out to be the barrier. You can get the farmers to grow it. The farmers grow it. They see good yields. They're very excited, and then they find out there's nowhere I can sell it, or I have to drive hours and hours to find somebody who's willing to buy the crop, and then they typically don't come back the next year. So I was making, this pitch. This is a wonderful crop. We can grow it in places where we don't have enough water to grow other things like corn. It'll help diversify crop rotations. It'll be good for food security. Why hadn't it expanded already when I came to make the case we should invest in plant breeding? Um, and the challenge was a lack of breeding activity. The vast majority of those 200,000 hectares of land were planted with a single variety called Huntsman, which had been developed in the early 1990s, actually at the University of Nebraska, where I am present. After that, the one breeder of proso millet had been hired away to go do something else. And so there was just no activity devoted to releasing new varieties. And so even though the efficiency with which the crop could produce grain from water was high, the yields were just so low it didn't make sense for farmers to grow in most cases. We developed a pitch which I basically just walked you through, for why it would be really valuable to invest in breeding this crop and why using a lot of the technologies that have been developed to keep pushing the frontiers of yield for crops like maize forward, we could go much faster and at much lower cost than to achieve some initial advances in proso millet breeding. As I mentioned, proso millet was domesticated in Northeast China and went through a series of bottlenecks as it moved from China west across Asia into Europe and then into the US. So one of the early things we found was that the proso millet varieties being grown in the US had extremely limited genetic diversity. They were almost impossible to distinguish when we were running genetic marker analysis on them. But there was a lot of diversity from around the globe, primarily from places like Eastern Europe, like China. And we were able to access a lot of that global diversity, as well as contributing to sequencing the first reference genome for this crop proso millet. Now, millet is a very confusing word. I think it's essentially the English word for a grain crop we don't have a name for. Uh, Different millets are not particularly related to each other any more so than corn is to sorghum is to teff. The other confusing thing is the same species can have multiple names. So what I call proso millet, other people might call broomcorn millet or common millet. Anyway, but we were able to contribute to sequencing the genome of this crop, generate large numbers of genetic markers, and use that to assist in our breeding strategy starting in 2015. We brought in a lot, as I said, a lot of diversity from around the world. We crossed it all together. We generated new varieties. We put them out in the field. And it was the most discouraging field season I've ever had. We walked through the field. Maybe 1 in 10 of the varieties even looked like a crop when we grew it in Nebraska and Colorado. There were, plots that had died, plots that hadn't flowered, things that flopped over. But 10% of them looked like a crop. We took those because we were able to use a very rapid cycling approach to generating new varieties where we could make crosses and advance them 4 to 5 generations in a single year and generate many, many new varieties. We were able to move through several cycles of selection and get past purging a lot of the material that was probably adaptive to other parts of the world, but terrible for growing in Nebraska and Colorado in the Western US. And what we were left with was a dramatically more diverse germplasm pool than any breeder in the US had had ever. So we started out, we brought in a lot of diversity. Our yields, our productivity, our adaptation dropped dramatically. And then we were able to come back up to the point where we caught up to what was currently on the market. And then we could very rapidly go past that because we were selecting from a much, much more diverse germplasm pool. Using genome-assisted breeding, this rapid cycling, testing many, many varieties. And okay, this is just walking you through the pipeline of how this breeding approach actually works. But what we achieved, and the first varieties went into pre-commercial release only 5 years after we started the company. We now have 4 varieties in the market. Were varieties that yielded up to 40% more grain than Huntsman, the standard variety that every farmer, almost every farmer in the US was growing. We are now in our second cycle of releases and are working with varieties that outperform even the first varieties that we released back in 2020. And we're not seeing any evidence of this rate of increase slowing down. Again, we're benefiting from our ability to bring in very, very diverse genetics so that we have more to select upon. When you talk to farmers, I mean, they will tell you what they care about is yield, at least in the US. And that is certainly true, but we have found that there are all these other little things that growers appreciate as well. We have thicker stalks on our proso plants, which allow the stubble left over after the harvest to catch more snow that brings more water down into the soil for the next crop the following year. We also found out that this particular variety, DLG 317, apparently tastes much better than any of the other varieties that were on the market. It wasn't something we'd even thought to select for. So now we're getting an increasing amount of interest, particularly from the East Asian community, which right now, even though proso millet is grown in the US, that community has been importing proso millet from China, from Korea, because those varieties taste better. And we coincidentally have found that we now have something that actually tastes good. And it's really quite powerful having the experience of seeing technologies that I learned about in school, technologies that I apply in my academic program, applied to actually making farmers' lives better and actually making more grain, more food for the world. These are just a couple of testimonials from customers who tried growing our varieties for the first time. So in this first part of the story, I wanted to demonstrate a bit about the different way that you make your case for doing something when you are working in the private sector, when you have to pursue private sector investment, but also the extremely large impact that plant science and breeding can have on the real world. This is our lead breeder within dryland genetics, a guy named Santosh Rajput. Uh, and he's had that impact. He came through, he did a PhD at UNL. We hired him at the company after he graduated. It's been really exciting. The other thing as I transition from this part of the talk to the next part is to emphasize, to mention, so dryland genetics achieved this impact, these increased yields, using conventional breeding, some marker-assisted selection, and genomic prediction. In 2014, when we were pitching the company, these were not tools that were widely used outside of the major seed companies. Today, they're very widely available. So as I transition into the last part of my talk, which I'm actually going to compress a lot because I think I've been talking slower than I thought I was, I want to talk about some of the new tools, the new technologies that I'm working on in my academic lab that I'm guessing a lot of you will have the chance to experience and employ either during your PhDs or later on in your professional careers. So one of these is genome editing, and people have been, again, very excited about this for a number of years. For a long time, genome editing has been limited by the capacity to do transformation, to introduce CRISPR and do edits. This was a study that came out almost a decade ago now, looking at if you could use genome editing to introduce even small numbers of alleles that you knew had a beneficial impact on a trait, say yield, say water use efficiency, say stress tolerance, how much faster your breeding program could progress than if you were using purely conventional breeding and genomic selection. It's a lot faster. On the other hand, if we're talking about almost any trait of interest to breeders, let's say yield, I don't think we have 20 edits that we know where we could actually know this is the DNA difference that will improve yield. I don't think we have 10. I don't think we have 5. We might have 1. We are still really bad at looking at a plant genome and saying, okay, these are the specific nucleotides that need to change if we want to change a trait in a desirable way. We can find the general region of the genome. We can find markers that can be used for genomic prediction. But we're really bad at this. We also know that more than half of total variation in traits is explained not by genetic variation within the sequence of genes, where we're at least a little bit better at understanding which DNA sequences produce which differences, but in the non-coding sequence between genes that determines when genes are turned off and on and how much they are expressed. So why is it? Why is it that we know? This is just a simple genome browser. We can see here is a gene. We've got exons, we have introns, we have UTRs. What we study is driven in large part by what we can see. We've gotten very good at, if you hand me the DNA of a plant I've never seen before and a budget, I can sequence it, I can assemble it, I can even run annotation, figure out where are all the genes in the genome of this plant that I've never seen before. And so because we can see these easily in genome browsers, this tends to be what we focus on as plant geneticists. We tend to focus on the coding sequence. But we know that there is lots of regulatory sequence that doesn't affect the protein, but affects that affects phenotype. We don't see it when we look at genes in genome browsers typically, and so we don't study it nearly as much as the coding sequence. So a graduate student in my lab set out to try and address this challenge, and how can we better see which parts of this non-coding sequence are actually regulatory, are actually things we should be studying as we try and understand how a genome specifies a plant. So this is Michael Tross. He graduated in May of this year. He's now over at Corteva as an AI scientist in their plant breeding unit. And he developed a transformer-based model to look at a gene and the non-coding sequence around a gene. He trained this transformer-based model to predict the pattern of gene expression across multiple tissues, um, actually 6 tissues in this case. And he didn't do this because we wanted to know which genes are expressed in which parts of the plant. We can figure that out directly. We can sample those parts, we can do RNA-seq, we can get gene expression. He did this because once he had trained his model to do this, he could then use an approach called saliency analysis to look at where in the region surrounding the gene were the DNA sequences that mattered in terms of how the model was able to make its predictions. So what you see here are some examples of individual genes, either in the corn or sorghum genome, and this is an even simpler genome browser because we built it ourselves in Matplotlib. But you see here the exons and the introns. And what you see here are the saliency scores. How much attention is the model paying to different parts of the DNA sequence? And you can see it pays relatively low attention to lots of the genome. And then there are spikes, regions where it pays a lot of attention. And those spikes tend to line up with other pieces of evidence we have that a particular part of the genome is functioning in a regulatory way. These can include conserved non-coding sequences, open chromatin regions. And he was able to take this model that was trained in corn and apply it to a relative, sorghum, without having to retrain the model. So what we think Michael has done, and this slide is a little bit out of date, the preprint is up on bioRxiv if you'd like to read more about it, is used an AI model that was trained to predict gene expression across tissues to figure out where are the non-coding regulatory regions in the genome that we should be paying attention to, both to understand how DNA sequence determines gene expression, but ultimately to find the DNA sequence differences we might want to introduce as part of a breeding program to accelerate rates of genetic gain. Let's see. Okay, so I think I've got about 15 minutes left, but I'm assuming that that should include time for questions.
Yes, please. Yeah.
Okay, so we are going to go very fast for about 5 minutes, and then we're going to call it done. So what I just described previously is once we have a candidate gene, we can look around that gene and figure out what are the parts of the DNA sequence that are of interest. But before we find a candidate, but first we need to find candidate genes. Typically, the way this happens today in a lot of plant genetics programs is through things like genome-wide association studies. So we start with some sort of diversity population. You see here, this is a diversity population of sorghum we grow in my lab. We generate both a bunch of genetic marker information for each plant, so we know what DNA differences there are between these plants, as well as a lot of trait measurements. And ultimately, what we're doing is simply comparing all the plants that have one allele, one genotype at a particular genetic marker to all the plants that have a different allele or a different genotype at the same genetic marker, and asking how different they are from each other. In the case of a genome-wide association study, you do this for hundreds of thousands or millions of markers, you end up with something like this, a Manhattan plot, where each point represents a test for a single DNA sequence. Yeah, we already saw this. While DNA sequencing is getting very, very cheap, the other half of that equation, the measuring the properties of plants, remains extremely expensive, extremely labor-intensive, extremely slow. There has been a huge investment, I think, around the world in technologies called high-throughput phenotyping technologies, which ultimately are just any technology you can use to substitute for people walking through a field with rulers and notebooks making measurements of plants. Yeah, okay. One of the things that is interesting about a lot of these high-throughput phenotyping technologies is that there's an extra step in the process. If I start out with this diversity panel in the field, and I go through with a ruler or notebook, I have some property I want to measure. I measure that, I write it down, maybe directly into a spreadsheet, maybe into a piece of paper gets typed into a spreadsheet, and then I can go and do my genetic analysis, agronomic recommendations, breeding decisions, what have you. With automated technologies, the vast majority of these, first you have either a human or a robot or a drone or a satellite that is collecting some sort of sensor data. In this case, a LiDAR point cloud, maybe it's a photo, maybe it's a hyperspectral data cube. And then there's an image processing step where we extract from this sensor data the measurements of the property we're particularly interested in. This means that once we have this sensor data, we can go back to the same field experiment over and over again and extract measurements of new traits without having to regrow the experiment, without having to send students into the field again to make additional sets of measurements. And that is changing the way that we do plant genetics and plant breeding because it makes it much more common and much cheaper to have high dimensional data sets where we've measured many, many different properties of the same plants, including properties we probably wouldn't measure if we had to go out and do it on our own. It would take too much time, it would take too much effort, it would cost too much money. But if we already have the sensors and we already have the algorithms, it's just very cheap to get more measurements of more things. Yeah. One of the things that we can do as we have more and more measurements of plants is we can try and replicate some of the things that still depend on human judgment today. If you look at the early stages of almost any breeding program, except maybe a couple of the biggest breeding programs for corn and soybeans, one of the first steps is selection by something we call the breeder's eye. A trained human being who has been doing this for years or decades walks through little plots and says, I think this one has the potential to have high yield, and this one does not. This can be Dr. Santosh Rajput, the lead breeder dryland genetics, but it can be people from anywhere in the world. And I'm citing two studies here that both demonstrate that in one case, actually with farmers rather than breeders, that farmers looking at these little plots can do a better job of predicting which ones will have high yield in which varieties will have high yield in subsequent large replicated yield trials than actually measuring the yield directly. So we have a human being who has trained their brain to look at many properties of a plant and predict which ones will perform well and which ones will perform poorly. And a PhD student in my lab, Hongyu Jin, was able to use some of these high dimensional data sets where we have measurements of many, many properties of plants in the field to try and replicate that. And he was able to essentially train a computer to look at other properties of a plant and predict which ones would have high and low yield in future years and future environments. Yeah, I was too ambitious in terms of how much I was going to talk about today. So we are going to skip through to the end. I'm going to thank you again for your time and attention. I'm going to thank the outstanding folks I get to work with here at UNL and my collaborators across the US and the world and the funding agencies, and I'd be happy to use the remaining time to take any questions you might have.
Yeah, Dr. James, thank you very much for this inspiring presentation. I think my wow at the beginning is now having all its sense at the end of the presentation, and kudos for that and the amazing things that have been done from academia and also the private sector. I think if you are a plant breeder on this call, you have the whole toolkit from the private sector to fundamental or basic research in academy and everything. So this is now time for a question. You can drop your question in the chat or kindly raise your hand and you ask your question. So maybe before we start getting questions, I think I have one question for you. This is in terms of you sharing tips with young breeders, and how do you mobilize such resources or such funding, especially for a private venture when you are dealing with a crop which is not even a well-known crop in your environment? How do you put things together to convince people to put in their money, because there is a high risk there. So how do you do that?
Yes, that's a very good question. I mean, I will say that I think academia prepares us surprisingly well for this. We don't realize that we're doing it. If you are writing a fellowship proposal, if you are writing a grant proposal, what you are doing is making a detailed case that there is something, it should be done, it's possible for it to be done, and you are the right person to do it. The big difference is that typically in academia, we're making long written proposals And for lots of fundraising, for lots of investment, you go into a room, you have maybe 5 slides, you have maybe 10 minutes to make the case, and then you're having an ongoing discussion with the people who may or may not want to invest in your company. So we learn a lot about how to make the case, but we have to adapt from trying to explain all the contingencies, all the scenarios, all the details, to what are those, core important points. What is the big problem? There are lots of big problems. We have no shortage of those. What is the potential solution? There are a good number of those. Why is it that it will work? And why are you the right team to do it? And it also works the other way. I think since I've started starting companies and making those pitches, I find that I'm a much more effective grant writer and a much more effective communicator in academia because I've had this pressure on me to how do you distill everything down to 5 sentences or 3 slides instead of only the long, complicated version.
Yeah, so there is a lot that can be learned both from the private sector and public. And if you combine both, then you become, you exploit the terrorists in there, and yeah, your hybrid becomes very
Exactly.
vigorous. Okay, all right, thanks. And any question in the chat? I think I still can see any question. Maybe they want me to ask more questions. Anyway, I will have a next one for you. I think when you start with the dryland genetic, as you mentioned, there are a lot of tools which were not widely used and which are now more, commonly used in many breeding programs around the world. From your experience and looking at the way things are moving, what do you think is the next game changer? I know you briefly talk about gene editing, but what else after the gene editing?
Yeah. Yeah. So I think it depends on which crops we're talking about. If we look at those, big national breeding programs, things like, corn and soybeans in the U.S., rice in China, wheat in India, I think a lot of it is that step beyond gene editing of actually identifying specific functional variants that can be directly introduced into the genome. The challenge in any breeding program is that once you are dealing with more than 2 or 3 loci, getting everything to recombine and trying to select good individuals becomes statistically quite challenging. We can sort through 10,000 individuals. We're not going to sort through millions. So in those national breeding programs, I think that is the the biggest thing we're going to see in the next 5 to 10 years. At the same time, though, I think that we have a need and there's a growing interest in diversifying crop mixes, investing in both different crops, but also more specialized local breeding efforts. And in many cases there, the thing that I think is going to change things dramatically are things like this phenotypic selection that I breezed through in about a minute there at the end of the talk, because genetic marker data is still expensive. If you try and pencil this out, I mean, it will be on the order of $20 a sample. In many cases, for small and specialized breeding programs, it is much cheaper and more effective to just generate lots of varieties, plant them out in the field. The big limitation then is access to skilled and effective breeders. It takes a lot of years to train a good breeder, and a lot of people try and they find out they're not very good at it. In the US, I mean, I saw many of my colleagues, you finish your PhD, you get hired as a breeder, you get sort of 5 to 7 years, and then they say, oh, nothing you picked is good, you're out. So if we can do a better job of helping breeders with machine learning tools that can work with photos of plants, with just hand measurements of plants, I think we can help a lot of these small and specialty and regional breeding programs catch up to the performance that the big programs are getting right now with genomic prediction, and we can do it a lot cheaper.
All right, thank you very much. There is one question in the chat. Let me read it from Rashidat. Hello, Dr. James. Okay,
Yeah.
can someone interested in only plant breeding be a graduate student in your laboratory at the University of Nebraska? This is more personal, but you can always address it if you want.
Yeah. I'm happy to address it. The students in my lab tend to not be plant breeders directly. I have several colleagues at UNL who have active plant breeding programs in crops like wheat and soybeans. The challenge that I face in my academic lab is I work primarily with corn, and in corn, the private sector companies are so advanced, we aren't able to develop competitive varieties at the university, and without competitive varieties, we don't have money that comes back to support a breeding program. So a lot of the research I do in my lab teaches people quantitative genetics tools, phenotyping tools that they might then go and use in a breeding program, but they're not conducting or learning breeding directly when they're grad students in my lab. But there are other programs that do that.
All right.
Other
Thank,
crops.
thank you. A question from Abel. Beautiful presentation. And how do you handle gene editing of quantitative traits such as droughts, if
Yes.
that is possible? Yeah.
Right now, I don't. I would very much like to, but there are probably a number of genes that have been linked directly to changes in drought tolerance. But even in those cases, we typically don't have the causal loci. The biggest, easiest thing to do with gene editing right now is to knock out a gene. If it was as easy as knocking out a gene and suddenly the plant is more drought tolerant, breeders would have found that. Genes get
Sure.
knocked out all the time through natural mutations that occur within breeding programs. My guess is that in order to achieve increases in things like drought tolerance, we're going to need to make more complicated changes, whether that is edits to the sequence of the protein, whether it's changes in the pattern of expression of the gene. And right now, we're not there. We need to be there, but we're not.
Maybe one last question from Innocent. Innocent, can you unmute yourself and ask quickly your question? There is more question in the chat, but I think Dr. James will address them as we move to the next session. Innocent. Okay, if not, I think it's time to wrap up this session. And thank you again. Thank you very much, Dr. James, for your enlightening presentation. And yeah, I will pass the floor to my colleague Zibrila for the next session. Thanks. And I hope you stay a little bit with us. I know you have a lot of commitment from private and public sector. So yeah, we'll be happy to have you for a few more minutes. Thank you.
I'll stick around and try and answer questions in the chat.
All right, thanks.